All right, so shall we start with the clustering part? Okay, so all the steps you have done so far, you've done a bit on the QC, and now we have cleaned your data. We looked at dimensionality reduction and so on. And one very important step that you need to do now is to identify the cell populations that you have in your data. This is a picture from a paper that I think it will be very nice to read from Wagner et al. It's basically on what determines a cell identity, right? So they put forward 4 things. So environmental stimuli, for instance, the developmental trajectory, where is that cell coming from? The cell cycle, which we talked a lot about also yesterday, spatial context, what's the environment in which this cell interacts, and so on. So all these things determine the cell identity that you somehow measure or try to capture with the transcriptome, right? So this is kind of the a very abstract view of the workflow that we're doing, right? So you have some mysterious cells. You do some measurements. So in our case, this is single cell RNA-seq. So you get a data matrix with cells and features, and these features are genes now, where you measure the expression of each gene. You somehow group the cells. And this is the clustering step, so we will talk about that. And then at the very end, you try to annotate these groups or populations to a known name or identity that you can relate to, right? So again, this is a bit in more detail. So we did QC, normalization, we need to do some feature selection, dimensionality reduction, and then the clustering step. So these are already covered so far, right? So this is what you just did this morning. We talked a bit about feature selection, but I'll reiterate that a little bit, and then we talk about clustering immediately. So this is the outline of what we will do. So why do you actually need to do feature selection to start with? There is something called the curse of dimensionality. And if you haven't heard of that, it's a bit counterintuitive. So you would think if I would measure more features about any entity, that I know more about it, which is not very true. Yeah. So here is an example where you look at cells in the PCA space. And on the left side, you're only looking at 500 genes. And then you see nice clusters of your data. Yeah. While on the right-hand side, if you look at 20,000 genes, you do see that the clusterings become a bit less clear. Like, for instance, the separation between gray and black here is very clear, while here it's a bit not very clear. Yeah. This is a bit difficult to think about, but the idea is that you basically, when you look at more features, you look at more noise as well, and that obscures the large or very clear distinctions in your populations. So there are 2 goals of removing genes that are only carrying technical noise. Let's call it that way, and that's basically to improve your signal-to-noise ratio. So I focus on the real signal in the data and ignore more the noisy signal. And also to reduce the computational complexity, because if you, let's say, have to calculate the distance between 2 cells, if you use only 500 genes, that's a lot faster than if you use 20,000 genes. Yeah? So with feature selection, this is one of the methods we looked at yesterday again, but I'll just reiterate. So your goal is to find genes that are very variable across your cells, right? The problem is, if you look at the relation between variance and the mean of the expression, there is an interdependence between those. So what you can do, and this is one of the very first approaches to do this, to plot the coefficient of variation, and that's the variance divided by the mean squared. And this is the mean. And then you see that there is some sort of a relationship in between. And so all the brownish color is all the genes. The blue colors are the spike ends. So then you can fit a function to the spike ends, right? So this is only technical variation, right? And anything that's a bit further away, so the purple colors are the genes that show variation above the technical variation that you would expect in your data, right? Now, yesterday it was mentioned already that spike ends are not very compatible with most of what you're doing in droplet-based methods. So one way to avoid this is to make the estimate of the red line based on all your genes. So what's the average variation in my data that I would expect without the spike ends, and then take the genes that again are further away from this line. Yeah. So you say, okay, I want a specific distance from this line, and then anything above this distance is variable. Yeah. Is this clear? Because we will look at these plots more in the presentation. Yeah. Another approach to do this is to use something dropout-based feature selection. And this is a method that relies on on this equation. So this equation comes from this guy. It's a bit of an old equation that looks at the dropout rates and reverse transcription, right? So you have the probability of the dropout is 1 minus S is the average expression of a gene, and then Km is some constant plus S. Yeah. So what's the intuition behind this is that if you have a control gene that's not variable, then you would expect some sort of a distribution like this, right? But if you would expect a gene that is variable across your cells, then you expect something like this, where there are a lot of cells that are around 0, and some cells have a higher expression. Yeah. So if you now plot the mean versus the dropout rate, so the dropout rate is how many cells have a 0 value over the total amount of cells. You basically, this is what you would expect. So this line, right? So you would see that a control would basically fit on this line, that the mean expression versus the dropout is predicted by the equation. While here you see that if I look at a gene in this case, the mean will be somewhere here, so it will be equal to the control, but the dropout will be much higher. Yeah. Then you can use this to pick basically the variable genes again. Again, you make a very similar plot. So again, dropouts, average, and then if you each dot is a gene, then you can fit a line to the average of all the genes. That's the blue line. And then if you say anything that's a bit further to the right of this line are the yellow dots, and these are, or the orange dots, and these are the variable genes based on the dropout selection. Yeah. You just heard about dimensionality reduction. So again, the same idea. So we very often reduce the dimensionality of the data using principal component analysis before we go into clustering. The reasons being very similar to why we do feature selection, right? So we expect the main components of the data to explain most of the variation. That's how we create them. And all the smaller components carry a lot of the small variation in the data, and then we don't care about it. We want to throw it away. So again, you created these plots right now. So this is the elbow plot. It's also called tree plot. So you plot these are all the principal components in your data. This is how much variance is explained by each component. You order them. So of course, the first component explains the most of the variation in your data, and then it keeps dropping. And you come up with a magical number of a cutoff of something. And then anything to the left you use for downstream analysis. Anything to the right you throw away. Yeah? Assuming that these mainly correspond to noise or variation that you're not interested in. It's easy to pick the first ones because they explain significant amount of your variation. It's easy to discard these ones because they explain very little of the variation. In the middle, it's called an elbow plot. It never looks really like a clear elbow, so it's not going like this and then flat. So you have to be creative. Okay, now we get to the clustering. There are many ways to do clustering. We'll not go through them all. So hierarchical clustering is one of the very basic methods. K-means clustering as well. You heard a lot about density-based clustering. We will not talk about this in the lecture, but there is a small example on this in the practical, just because it's not used as often in single-cell RNA-seq data now. And we'll talk about graph-based clustering methods. So these are the ones that we will go through. Keep in mind, I'll use very basic examples to explain these, and this one as well, and then we will talk about single-cell specific methods at the very end. Yeah. Okay. Who knows what hierarchical clustering is? Okay, cool. So just to refresh your memory a little bit. So these are two clusterings of the data. Who thinks the one on the left is better than the one on the right? Who thinks the one on the right is better? So if you said the one on the left is better, why do you think so? Why is it better, actually? What? Sorry? Yes, a bit simpler terms than silhouette.
The red lines don't touch.
The red lines don't touch, okay. Some other criteria?
The dots inside the circuit are more close than...
Yeah, So this is indeed what I was getting at. So basically distances between the dots within a circle are smaller than distances between the dots across the circles, right? So that's how you can explain this also to the algorithm, right? And that's basically what most of the algorithms try to do, right? So you try to measure some variance between your points within a cluster, some variance across the clusters, and then your goal is to minimize this function, right? That all the variances within the clusters are smaller than the variances across the clusters. Yeah. So this is kind of your objective function. Okay, so how does it work for hierarchical clustering? So here I have a toy example where I have 8 points, and they are lying in 2 dimensions, no matter what these dimensions are for now. And we want to cluster them, right? So in a hierarchical or agglomerative clustering, what you basically do is you start by joining the 2 most similar cells, right? So you calculate all the pairwise distances between your points, and then you take the smallest. You join them, and the height of this thing on the dendrogram is basically the distance between the 2 points. And then you go for you go for the next closest object, and in this case it's 5 and 8. You join them, put the distance on the height. Then you continue, and now we basically join the cluster of 4 and 2 with the data point of 3, because they are the closest. Then we do the same again, so this cluster now is joined with 0.7, and then you basically continue until everything is in one cluster, and then you have finished. Yeah. Then if you say, okay, I'm interested in 2 clusters, then you can cut this dendrogram here. That gives you 2 branches, and that would basically be this cut in your data. Yeah. So I kind of withheld some information from you. So what more information do you need actually to do this, right? To build this dendrogram? What do you need to define extra? Yes. So there are 2 things that you need to define. So first of all, how do you measure distances between your objects? And how do you measure distances between clusters as well? Because at some point I joined point 3 to this cluster, but how do I measure the distance between a point and the cluster, or 2 clusters? Yeah. So let's start with the distance between the clusters. So we have those 2 clusters, and we want to measure the distance. And there are different ways to do this. So these are 3 common ways. So you have single linkage, complete linkage, and average linkage. So in single linkage, you basically, in all of them, you calculate all the pairwise distances between the 2 clusters. So between all the points in one cluster and all the points in the other cluster. Single linkage says, well, just take the shortest. Complete linkage, take the furthest. And average, just take the average distance. Yeah. Easy. Okay, so let's have a small exercise. So I have the points here on the left, so we focus on this for now, and I have already grouped them in these 4 groups. So I stop at this level, and I want you to think, like, if I want to now join them based on complete linkage, which 2 groups would be joined? So should I now group this with this, or this with this, or this with this one? This and this. Yes, that's true. Does everyone get why that is the case or not? Okay, let's try to walk through it a bit. So you want to calculate all the distances, let's say, from this cluster to the others, right? So we're using complete linkage. So we always take the farthest distance. So the farthest distance between this and this one would be here. This one and this one would be this. And this one and this one happens to be this one, right? So once you have calculated all 3, then you take the minimum of those. Right? So you only look at the maximum when you calculate the pairwise distances, but then you select the minimum to join. Then this is the shortest one, then I end up choosing those. Yeah. Okay, let's do the same now for shortest distance. So again, I want to join them based on the single linkage. So this is the shortest distance between those. Shortest distance here, shortest distance there. Out of the 3, this is the smallest, so then I end up joining those 2 clusters. Yeah. How would it look like for the one on the right? So if I do it with single linkage, any guess? Okay. So it will look something weird like this. So the yellow one, like this, and this one will be like this. If you do complete linkage, then you make 2 halves of the data. Yeah. So the choice of the linkage really makes a huge difference on how you would cluster. And keep in mind that a general rule is that if you do single linkage, you end up with long and loose clusters because you keep like moving one point at a time, and then you keep growing like very long structures. Well, if you do complete linkage, then you always take kind of outliers into account, right? So you end up with bigger clusters. Yeah. Okay, the other thing we have to think about are distances between the data points, right? So we already talked about Euclidean distance in the morning. You can also use Pearson correlation or mixed Pearson correlation. So basically, it depends on what do you really want to capture. So if you do Euclidean distance, then you're really looking for exact matches. So in this toy example, then green and blue will have the smallest distance, right? Because their shapes match quite well. And although the pattern of the orange line is kind of similar, the Euclidean distance will be larger between any of those two and the orange one. If you do 1 minus correlation, then you don't really care about the amplitude. Then the distance between green and blue will be identical to the distance between green and orange, for instance, yeah? Or very close by, because you only look at the trend of the data. And if you also take the absolute of the correlation, so 1 minus the absolute of the correlation, then you also take like inverse relations into account. So the distance between blue and pink is also now comparable to the distance between blue and green. Yeah. Okay, so this was hierarchical clustering. I think it's relatively simple, and it builds on the idea of building your cluster as a tree, right? The other type of methods is, K-means is a good example where you try to look for prototypes in your data. Yeah. So the way it works is that you've got your data. You start by selecting 2 random prototypes, and there are different ways to do this. So you can randomly pick any 2 points in your data. And I say 2 because you have to specify before you start what K is, right? How many clusters do I want? So if you say I want 2 clusters, then you need to start with 2 prototypes. You can pick them as you wish. So you can pick them as random points in the space, or you can just take 2 random cells or objects that you have, or whatever clever way that you want to think about. And what you do is you basically calculate the distance between each point and each prototype. And then let's say this one is closer to the blue than to the red, then I assign it to the blue point. Yeah. So if you do this, then some of your points will be assigned to blue, some of them will be assigned to the red one. Yeah. Then you update your prototype by calculating the average of these points. So now my prototypes have moved because all these points were assigned to blue. Then the prototype of blue is now was here, now it moved here, and the same for red. It was here, now it moved here. Yeah. Then you do the same again. Then you ignore the blue and the red, and you calculate the distances to the prototypes. So again, this one will now have a closer distance to this one than to this one. Then you update your labels. Yeah. And you do that again until there are no changes, basically, right? So now I updated, my prototype has changed, but the assignment of the points has not changed. So then I stop. Yeah. Then I end up with these clusterings. Yeah. Any questions?
Yeah, about the hierarchical. So here we choose the prototype, but how it's going on there?
So in the dendrogram, when I said like you need 2 clusters, and then you cut the dendrogram where at the height that gives you 2 branches. If you say I want 3, then you need to go down a little bit until the tree breaks into 3, and then that's the cut of your data. And there are different ways to cut. So now I only showed you one horizontal line to cut, but there are ways to cut the tree in a very funky way. Yeah.
But you still specify clusters.
Yeah, we'll get that to the end. So nobody should sell you a method that does not specify the number of clusters. At some point, you make a choice about the number of clusters. Yeah, we'll see. It's a bit easier to stay away from it in graph-based clustering because it's a bit farther away. While in K-means and the hierarchical clustering, you set it like you say, I want this many clusters. It might be named something else, like, I don't know, a distance or a similarity between your objects or whatever. It's at the end a selection on the number of clusters. Yeah. You had a okay. There are some limitations or problems with k-means. So if you follow this procedure, you always end up with round clusters. Yeah. So k-means will always favor that you have round shapes. And that's a bit problematic because, well, life has a bit more complex structures. So here, for instance, this is a famous data set called banana data set where these are the two classes that you have. And if you do k-means, you get something like this, which is obviously wrong. These are the true classes, right? Same here, for instance, if you have two clusters within each other, like this ring structure, this is the true labeling. K-means will give you something like this. Yeah. So you have to really keep that in mind, that k-means has a specific assumption about the shape of your data. So again, if you cluster data with k-means on the TSNE or the UMAP space, keep in mind that if you have a round cluster, that's easy to define. If you have a long shape, then k-means cannot resolve that. Yeah. And again, you need to specify k, but that's also the case for every other clustering method. Yeah. It's here just a very hard decision on what do I want to get out of the data. Conversions is a bit of an issue, but you can forget about that for now. Okay, so let's talk about graph-based clustering. So just before we start, what are graphs? So graphs are networks that have nodes and edges. So edges connect basically multiple nodes, and there are many definitions of graphs. We will talk mainly about a simple case. So we don't have arrows here, for instance. So this is not a directed graph. This is a non-directed graph. This one, for instance, doesn't have weights, so all the edges kind of equal, but you can also have weights on the edges. So keep that in mind. But the basic idea of having nodes and edges connecting them is simple. And the goal of graph-based clustering is to find what's called communities or subclusters in the graph. So parts of the graphs that are very well connected compared to other parts. Yeah. In the single cell field, there are mainly two types of graphs that people talk about. So there is the k nearest neighbor and the shared nearest neighbor. They are very similar, but still different. So in the k nearest neighbor, you build a graph where 2 points are connected if let's say I'll connect P to Q, if Q is among the k smallest distances to P, right? So I calculate all the pairwise distances, and then if k is 30, then I take the closest 30 points, and I connect them to my cell that I started from. Yeah. The shared nearest neighbor is a weighted graph where you say, okay, I have two cells, and I'll connect them with an edge, and the weight of that edge depends on how many other shared points they have, right? So if those two nodes are only connected to each other and they don't have any shared neighbors, then the weight of this link is very small. But if they fall in a neighborhood where there are also many indirect ways to get in between, then the weight of this edge will be higher. How do we actually represent a graph? Because you cannot put this directly in the computer, right? So you need to represent it somehow. So you can either use an adjacency matrix, and this ignores the weights, right? So you only look like this node is connected to this node. So then A and B has a 1 here because there is an edge. And A and F are not connected, so there is no edge here. Yeah, but you can also take the weights into account, and this is a weighted matrix. So between A and B, I have an edge, and the strength of this edge is 0.8. Again, reiterating, so graph clustering is basically also called community detection. So communities are clusters, and these are groups of nodes that have higher probability of being connected to each other than being connected to other nodes in the graph. And the community detection is, well, the goal is basically to find these groups of nodes or communities that share more edges in between than they share with other communities. Yeah. The way to do this, the way to cluster a graph is to use graph cuts. Yeah. So you basically, a graph cut is just a line that would cut the graph into 2 parts or more. And the size of the cut is basically how many edges do you have to cut. So the size of this cut is 1, the size of this cut is 2. Yeah, if we talk about binary graphs. The smallest cut is not usually the most optimal one. Like you see here, if I cut the graph here, then I end up with this and this one, which does not satisfy the concept I mentioned in the previous slide. Like there are not many more connections here than it would be between between these two, for instance. While if you cut it here, it seems there are more connections between this group and more connections between this group and less in between connections. Yeah. So we need to define this graph cut in a way that we can optimize. So one way to do this is to use something called the normalized cut. It's not very complex, but it's this equation. So this is the normalized cut between two groupings of the data. And what you do is you calculate the size of the cut. So again, how many edges did you have to go through? And you divide that by the size of one group. And on the other side, again, the size of the cut divided by the size of the other group. Yeah? And then you want this to be minimum, basically. So let's look at an example, because I think that's a bit easier. And here we do it on a weighted graph, not a binary graph. So the cut S and T in this case with the red line basically goes through those two edges. So then you just sum the weight. So 0.1 plus 0.2 gives you 0.3. Yeah. Then the volume of S, which is the one on the left here, is basically the size of the cut, so 0.3 that we just calculated, plus 0.8, 0.6, 0.8 gives you this number. Yeah. You can do the same for the T side, so the other side of the graph, and then you get and you end up with this number. And then if you use the equation from the previous slide, then you get 0.24. Yeah. If you do the same for this cut, which obviously looks like a bad cut of the graph, then you get something like 1.62. Then obviously this is smaller than this one, so you say, okay, this is a better cut. Yeah. Well, there is one problem. This is NP hard. That means that you cannot solve it in a reasonable amount of time, right? So you will need to try all the possible ways to cut and then come up with a minimum, which, okay, might be possible in this case, which I think is already difficult, but think about a graph of 5,000 cells, then it becomes intractable. Yeah. So you need to come up with a heuristic, and that's basically a way that's not solving it optimally, but good enough. And there are many ways to do this. The most common one that you probably are using already is Leiden, and that's implemented in a lot of the single cell packages. There's also Markov clustering that's being used. I think if you know, like the group of Sten Linnerson, for instance, and Karolinska uses this one often now. But there are many other ways to do this. Okay, so talking about the single cell methods, there's, again, like yesterday, there are a lot of methods to do the clustering. This is a nice review paper from Martin Himberg's group, listing just some of the tools, and so I highly advise that you have a look at this one, just to also understand a bit what are the challenges in clustering that people developing the methods are facing. I always like to show this one. This is a place where you can look at any package that's doing anything with single cell, and there are already 118 methods to do clustering on single cell RNA-seq data, even more for visualization, but a lot for everything. Yeah, so just to show you that it's a bit difficult. I'll only show you 2 today out of the 118. So SC3, it's called single cell consensus clustering. It's if you use the single cell experiment package, that's what you would do by default. I'll explain it. So it has these 5 simple steps, actually. So you start from your data matrix. You do some filtering on the genes, so removing genes that are not informative and so on. So that's simple. Then you calculate the distances between your cells using 3 different methods. This sounds a bit strange, and to me, at least, it's not. I don't know what the motivation would be to calculate different distances between the cells, other than we don't know what's an optimal choice, right? So what they do here is they calculate 3 different distances between the cells. So you end up with these distance matrices that have cells and cells. So it gives you the pairwise distance between all the cells using 3 different approaches. Yeah. Then for each of these matrices, you do dimensionality reduction with 2 methods, so PCA and Laplacian or spectral. Yeah. So now 3 becomes 6 because on each one I applied dimensionality reduction twice. Yeah. So now I have 6 matrices, and then for different numbers of components. So D here is like the first principal component, or if I take 2, or if I take 3, if I take 4, I take 5, and so on until a certain number. And for each one of those, you do clustering with k-means. Still following? Yeah. Now we ended up with a lot of clusterings because you do now 6 times how many components that you choose. So if you choose 20, then you're doing 6 by 20 clusterings. Yeah, but it's with k-means, so it's okay. And then you do consensus clustering, and consensus is basically for each pair of cells you check, are they clustered in all these matrices? If yes, then you give it a 1, and if they are not in all, you give it a 0. And yeah, there is of course a threshold if you say, okay, it's clustered in 80% or 90%. Forget that. You end up with this consensus clustering, and then you do hierarchical clustering on this one. Then you end up with the final clustering of your data. Yeah, so it sounds a bit yeah, complex, but it's in my view, it's just a way to go over all these choices. So now you're not really making choices, you're just using them all, and wherever they agree, you consider this a good clustering. Yeah. Okay, here I just wrote the steps so to make it easier for you. Okay, let's go to what happens in Seurat. So Seurat does something a bit different. It's based on other methods, by the way, so they didn't come up with this. You can read this initial bioinformatics paper, and then the main approach was used in Phenograph as well, which was clustering approach mainly for mass cytometry data. So what you do is you basically construct your k-nearest neighbor graph, right? Based on Euclidean distances on the PCA space. So first you reduce the dimensionality of your data with PCA. You use the retained principal components to calculate, get to a k and n graph. Then you refine this graph based on the Jacquard distance. So this is the toy example they gave in the Phenograph paper. So now I have two toy clusters. So one that looks like the banana shape and one is very concise here. Right? If you make a k and n graph with k equal 5, then you see that you break your big cluster even in smaller chunks because you're only looking at 5 neighbors. Yeah. So indeed, you can separate this one from this one, but you can also break this one into many smaller clusters. If you increase k, if you look at more neighbors, so k equal 10, then you join the 2 clusters. Now you cannot tell the difference between the 2. So what you can do with this Jacquard is that you can say, okay, I calculate the k equals 10, and then I refine this cluster using the Jacquard index. And the Jacquard index is how many shared neighbors are there between each pair of nodes. So in this graph, for instance, these 2 nodes here, I don't know if you can see that, do not share a lot of neighbors, right? So then I remove this one. While in this case, any pair of nodes within this small cluster will share many other neighbors as well, then I keep these, right? And then what you see here is that this resolves the true clustering of the data. Yeah? Okay, after you have created this improved graph, then you need to cut it somewhere, right? So, and what they do here is I told you about the Louvain is the heuristic, and it's a way to cut the graph by optimizing what's called modularity. An easy way to think about this is that if you cut the graph at every edge, so remove one edge at a time, and then you check if my graph is now split into 2. That means that this edge has a high betweenness. It's an important highway, right? So if I cut this highway, I cannot move between the 2 communities. Then it's likely that this edge exists at a boundary between the 2 groupings. Yeah. But never mind, so you can optimize this value q, and that will give you a number of clusters at the end. Yeah. The good thing about this one is that you don't really need to, in principle, define the number of clusters. But again, there are a lot of choices that you have made here, starting from the k, for instance, that you look at. So that determines how far are you looking actually in your community, right? So if you look much further away, then you, as you see here, you will end up with less number of clusters. If you look very, very closely, then you end up with a large number of clusters. Yeah. So it's an indirect way to define your clusters. Yeah? And I think, I don't know, did they change this in Seurat? It's called the resolution parameter. Yeah, it's still the same. So you make this resolution higher, you end up with a larger number of clusters. You make it smaller, you end up with a smaller number. Okay, so I have a few more slides on things that you need to think about when you're doing clustering. So how do you compare clusterings, for instance? A very famous way is called adjusted rand index. And it basically, it's relatively simple. So here I have a set S that has n elements, and I group them twice once I ended up with X groups and once I ended up with Y groups, you can build a confusion matrix or a contingency table that gives you the overlap between these groups. So how many elements in Y2 are also in X1, for instance? Yeah, it's easy to calculate. I think even in R there is a function to give you a confusion matrix immediately. And then you apply this not so complex equation to get the adjacent rand index. So it's a value of between 0 and 1. So if you have 1, that means that these clusterings are identical to each other. If it's 0, that means that these clusterings are vastly different from each other. Yeah. This is a nice a benchmark paper from Mark Robinson on many clustering methods for single cell RNA-seq data. So you see here SC3, Seurat, and many others. The different matrices correspond to different ways to filter genes. So this is based on high expression. This is based on highly variable genes. This is based on the dropout rate. Yeah. Dark blue means high adjusted rand index. Yellow means low adjusted rand index. So blue is better, dark blue is better. And you see here, for instance, there is more blue in these matrices compared to this one. So that means that using these approaches to filter genes is probably a bit better than this one in general on these data sets. It also seems that on this side of the matrix, you have more darker colors. So that means that these methods are doing a little better than those ones, for instance. So, and again, if you look here, it seems SC3 and Seurat are doing quite okay. I think in the paper, they make a recommendation on like if you cluster few cells, SC3 performs well. I think few is less than 5,000 or something. And larger Seurat works well. Yeah, I don't know. I think they both work quite okay on the data. Just don't use stuff here. Like flow sum, for instance, works very well for flow cytometry data. Apparently, it doesn't work very well for single cell RNA-seq. T-scan is, because I mentioned at the beginning, you will do something with k-means on TSNE space. I think T-scan is one of those methods. So you see, it's not too bad, but in some cases, it's still doing worse than these methods, for instance. This is how these methods are similar to each other based on the underlying methods. Like the top thing is what type of dimensionality reduction method they use, and red is PCA. So you see PCA is quite common. Some of them use TSNE, for instance. This is based on what clustering method they are using. So is it a graph-based or hierarchical or whatever? So you see quite some variation. And what type of input data they work on. Is it the raw or the log normalized data or something else? And it seems that the majority work with the log normalized data. Some can handle raw data as well. Yeah. So these are the things to think about if you're choosing which method should I use. Okay, a few things to at the end. So think about clustering. It's very subjective. Like from a computer science of view, it's a ill-defined problem. Like you cannot give it to a very powerful computer to solve. Like there is nothing like this because it's always subjective. You make a lot of choices about what similarity measure do I use, what algorithm do I use. There is no ground truth, right? There is no ground truth at the end. If there is a ground truth, we should do classification. So the problem is we don't have a ground truth. You have some cells and you want to look if there is anything interesting in them, and then you define them as this population, right? If you knew beforehand that it should be this population, then you shouldn't cluster. A lot of things become reality while they're very subjective, right? So people make choices and they define things, and then after a while, this becomes the reality that you have to adhere to, while they have also made a lot of choices. So look at the choices people have made. So if you compare your clusterings, for instance, to another paper, look how they have arrived at these clusterings. Because if you're using a different method, it's likely that you end up with something also different. And it's really a process. So think a lot about validation. So how do I validate my clusters? And yeah, I'll get back to the validation at the very end. How many clusters do you have in your data? This is, again, very difficult to decide. You need to play around with the parameters that you have, right? So again, in Seurat, you have this resolution parameter. Do play around with this parameter to see how much they change, for instance. Is it very sensitive, the clustering that you got? So you're very happy that you isolated your cells of interest in one group, but was it just this luck that you got there, or is it really a group that you can always define no matter what method that you try? A very common way is to look at differentially expressed genes. So if you define a cluster, then compared to other clusters, are the differentially expressed genes meaningful? Do they make sense or not? One common way to assess computationally if you converge it a little bit is to use bootstrapping. So what you do traditionally is you take your data, you cluster, and then you end up with the clusterings. What you can do is you can subsample your data, take a smaller set of cells, cluster them, and then repeat this many, many times, and then check if every time you cluster using a slightly smaller set of the cells, do I always get the same structure in my data or not? Yeah. And if that is the case, that means that this clustering or structure in your data is relatively stable. Yeah. Again, it doesn't say much about the biological interpretation of these clusters. It just says that computationally, I can reproduce this. It's not so super sensitive to the input data. Always check your QC data, as was mentioned yesterday. So what you don't want is that your clusters are defined only based on the technical variation in your data, that the clusters map to groups that have high gene counts or low gene counts, or high mitochondrial content and low mitochondrial content. That's something you don't want. So always try to make these plots, and they're relatively simple to make, where at the top you put all the metadata that you have, right? So this is the donors I have, this is the cell types, this is the log 10 of the green one, is the log 10 of the total features, so the number of genes or UMIs. And then you don't want something, in this case, for instance, this cluster has a relatively lower count than this one. So these are things that you should be worried about. How to assign cell identity. This is an art, and this is probably where you will spend all your time in the analysis, so forget about anything else. Annotating the clusters takes an awful lot of time, because what you really want to do is for each cluster, define the differentially expressed genes or overlay known markers, and then trying to decide, okay, this really overlaps with what I expect or what I know or not. Yeah, I don't know how to help you there. There is something, so there are these two databases that collect marker genes. So I think that's already nice if you don't know anything about the system, but in reality, you always know something about the cells. So I'm not sure if it's useful, but I think they are quite nice resources. They are a bit biased to mouse and humans. So if you also do other organisms, you're on your own. Do consider differences between markers. Like if you have a human sample from the same tissue, and then there is another paper that did exactly the same tissue from mouse, be a bit careful when you look at markers, because markers are preserved, but some of them are not. Yeah. Again, these are the main challenges, I think, in clustering, that we actually don't know how to define the cell types per se. It's a bit of a difficult decision. What is a cell type? What's a cell state? What's a cell population? Blah, blah, blah. The number of clusters is a bit of an annoying thing that you always have to play around with. Scalability seems a bit of an issue. So most of the graph-based methods don't scale very well. Like if you have a data set of 1 million cells or something, then it becomes very, very difficult to do graph-based clustering on this data. There are, like if you know ScanPy, for instance, it's a package from Munich from Fabian Theis. It does handle large numbers of cells quite well. So if that's the scale that you're working at, do look at methods that scale better. Because you will be really surprised how quickly most of the stuff we use break at a large number of cells. And again, for validation, rethink again about the cell identity. So it's not only coming from the transcriptome that you have observed, right? So if you would validate, try to think about other approaches. So it's very common now to look at the spatial context with the spatial mapping techniques. And I think it's a very good way to make sure that the populations that you have defined are also important in the context where they come from. But there are also other approaches. So if you're doing patch-seq, right? So if you're doing brain patch-seq, so then you can also look at recordings of the neurons, for instance. So try to always think about a different independent approach to validate the transcriptome-based clusters that you get. That's it. I hope we now covered the empty boxes in this plot. In the practical, I think that's after lunch, we do a bit on the feature selection with picking highly variable genes. Again, the dimensionality reduction to make these scree plots, as you saw. So I think you only did it with the Seurat in this morning, so now we just do it manually. So if you have any PCA or dimensionality reduction, how to look at the variance in the components, we do an example with hierarchical clustering, and I show you a couple of different distances and linkage methods that you can play around with. As I said, I have a small example where how can you apply k-means to a TSNE embedding, or try to put a UMAP in it and try again. And then I have a graph-based clustering example with different values for k. I did not put the one from Seurat because that's very easy to run. If you just look at the vignette, you will be able to run it. In this one, you kind of do things a bit separately just to understand what happens in each step. And that's it. A bunch of resources. Enjoy lunch.